Welcome to the Wright Science Daily Revision Boost. Today's Biology Blast, we're going to look at food security and selective breeding. So for those of you doing GCSE biology, you need to know that food security is the ability of human populations to access affordable food of sufficient quality and quantity. Now, there are several factors that will threaten food security. Firstly, the increasing population of humans. Secondly, changing diets, so that means it's more varied and more meat is required. Thirdly, climate change, so droughts, expanding deserts, etc. And finally, new pathogens and pests are evolving. So in order to maximise production, there's a few different stances we can take here. First one is actually taking steps to maximise photosynthesis. Using glass houses that control light intensity, temperature and water. We can use fertilisers to replace lost minerals. We can remove competition and pests through the use of herbicides, fungicides and insecticides. And we can also use pest-resistant varieties of crops or use high-yield crops. There are 4 types of chemical we need to know the names and what they do for. Fungicides kill fungi, pesticides kill pests, insecticides kill insects, and the only one that breaks that little standard is herbicides, which kill weeds. They don't kill herbs. What we actually have at our disposal are 2 different methods of farming: intensive farming or organic farming. When we're talking about intensive farming, this is using techniques to produce the maximum yield of food from the minimum area of land. So in order to carry this out, we will use chemical fertilizers and pesticides. We're going to use different techniques to maximize animal growth rates. We're going to use a lot more machinery rather than manual labor. Second option, the organic farming. This uses natural methods of producing crops and animals, avoiding the use of chemicals. One phrase we do need to know the meaning of is sustainable food production, and that just means we're producing food in ways that can be continued indefinitely. Sustainable farming techniques are going to be minimising the use of fertilisers and pesticides, and the way they do that is by using manure instead of artificial fertilisers. We'll use crop rotation to give the soil a chance to recover and prevent crop pest build-up. We'll also use pest and disease resistant varieties of crop which we've produced through gene technology, and we'll use biological control instead of pesticides. When we're talking about biological control, this is where we use natural predators of the pest species to control their numbers. So we breed large numbers of the predator and then release them onto the crop with the goal that they will eat the pest. 2 key problems with this though. Number 1, the predators may decide to leave the area, and secondly, they're living organisms. They may just eat other organisms and leave the pest perfectly happy and eating your crops. Another key technique we use is fish farming. So fish are a really valuable source of protein to us, but overfishing has led to population reductions for some of the species. So what we actually have done is introduce fishing quotas that limit the number and types of fish that can be caught, and we can use fish farms which keep fish in cages in seas and rivers, which protects them from predators and makes them much easier to catch because you just go to the cage and take them out. This also gives the wild populations a chance to recover. Couple of downsides though. Number 1, disease can spread quickly as they're very close together, and secondly, the risk of diseases spreading to wild populations is present because the wild populations can come right up to the edge of the cage. The last technique we need to know about is hydroponics. So this is where, as opposed to growing plants in soil, we grow them in a solution that contains the minerals they need. So this means we can actually stack plants up above each other to maximise the space that we've got to grow plants. One key process that everyone needs to know, whether you're doing combined or the GCSE biology, is selective breeding. So selective breeding is just the process by which we as humans breed other animals or plants together for particular characteristics. So things like a high yield or disease resistance. So the way we do this 5-step process: number 1, we identify the characteristic that we desire. We will then select the parents with high levels of that characteristic, breed them together, and then from their offspring we select the best ones and then we breed them again, and we will continue this for many generations. There are some big downsides here. Number one, it reduces variation within our gene pool because it's reducing the number of alleles present. So this means they're potentially more susceptible to diseases. And secondly, it increases the risk of inheriting a genetic disease. One mathematical skill we need to understand with regards to this is percentiles. So percentiles place data in ranked positions. So the median is your 50th percentile. The top 25% of results are at or above the 75th percentile. So that if we had an example where there were 200 sheep, our median would be where sheep 100 is. So in the set of data there, that would be at 40 kilograms. Whereas if we were looking for the 75th percentile, that would be where the 175th sheep is. So that would be 44 kilograms. In today's chemistry check, we're going to be looking at the GCSE chemistry only topics of making fertilisers in the Haber process. Hopefully we remember that essential elements are the elements that plants require for growth, and they're nitrogen, phosphorus and potassium. So if we don't have enough, we see mineral deficiency symptoms, which allow us to identify which mineral is lacking. So if there's a lack of nitrogen, we get yellow leaves and poor growth. A lack of phosphorus is discoloured leaves and poor root growth, and a lack of potassium is poor fruit growth and discoloured leaves. The reason we use fertilisers is that they replace the elements that plants use as they grow. Key thing about our fertilisers is that they must be soluble in water so that it dissolves and is then absorbed through the roots of the plant. So nitrogen is taken up as nitrate ions, which is NO3-, or ammonium ions, NH4+. Phosphorus is taken up as phosphate ions, PO4-3-, and potassium taken up as potassium ions, K+. The type of fertiliser we tend to use is NPK fertiliser, and NPK just stands for our three essential elements: nitrogen, phosphorus and potassium, and we get them in different ratios depending on which fertiliser we pick. The process we're going to use to make ammonia is the Haber process, and we make it from nitrogen and hydrogen in a reversible reaction, and you've got the balance symbol equation on the right there. So the raw materials for the Haber process are air, natural gas and steam. So nitrogen is made from the fractional distillation of liquefied air, and hydrogen is made by reacting natural gas with steam. Do remember that it's not just one fertiliser we make. Fertilisers come in a range of different chemical compounds, which I've given you on the screen there. Key thing they could ask you to do is work out the number of different elements that make up each one, or the number of different atoms. They could ask you which one would be the best one for replacing nitrogen, in which case find the one with the highest number of nitrogen atoms, and that's your pick. So when it comes to making fertilisers, the way we do this is by a neutralisation reaction between an acid and an alkali. So acid plus alkali makes a salt and water, and do remember when we're asked to name salts, then the second part of the name comes from the acid. So hydrochloric acid makes a chloride, sulfuric makes a sulfate, nitric makes a nitrate, and phosphoric makes a phosphate. And the first part of the name is just the name of the metal from your alkali. So if they would give you a question, ask you what acid and what alkali we use, then you can suggest whatever metal hydroxide as your alkali, and then look at the ending of your salt and then pick the right acid. So the method we use for this is just the titration. So you fill your burette with acid, use a volumetric pipette to measure 25 centimeters cubed of your alkali, which we put in a conical flask, add a few drops of indicator like phenolphthalein, record your start volume, add your acid while swirling the flask until your indicator changes colour. Record your end volume, and then you repeat that until you obtain those concordant readings which are within 0.1 centimeters cubed of one another. Once you've done that, add activated charcoal because that attracts the phenolphthalein, filter the solution, and then evaporate off the solution just to leave your fertiliser crystals, which you can dry in an oven. When we think about how to make chemicals in industry, we have a choice of two processes, either continuous or batch. So if we need bulk chemicals in large amounts, then we use continuous process. If we want specialty chemicals like pharmaceutical drugs, which are only needed in small quantities, then we use batch process. In the table here, we've got a comparison between batch process and continuous process. So what I'd suggest you do is learn a minimum of 3 of them, because that way if they ask you to do a comparison between the 2 processes, even if they gave you one in the question, you've still got 2 others that you can include in your answer. If they ask you to select which process will be best for making a particular type of chemical, you then have 3 ways to justify that choice. For those of you doing the higher tier GCSE chemistry, you need to know the intricacies of the Haber process. So as we've said, it takes nitrogen and hydrogen as gases and then it converts them into ammonia. So to do this we need 3 conditions: 450 degrees Celsius, 200 atmospheres and an ion catalyst. So the reason we use 450 degrees Celsius is that when we're looking at this reaction, our forward reaction is exothermic and the backward reaction is endothermic. So if we increase the temperature, our equilibrium position moves to the left, which would decrease the yield of ammonia. So the lower the temperature, the higher the yield. However, the higher the temperature, the faster the rate of reaction. So 450 is the compromise between the rate and the yield. When it comes to the choice of 200 atmospheres as the pressure, again it's a compromise here. So if we look at our balance symbol equation, we can see we've got 4 moles of gas on the left and 2 moles of gas on the right. So if we increase the pressure, the equilibrium position moves to the right and the yield increases because that's where fewer moles of gas are. However, we don't want to increase the pressure endlessly, because what we actually find here is that the higher the pressure, the greater the risks and the more expensive the equipment will be. So 200 is our compromise, high yield without the extravagant costs and risks. The last thing to consider is that we're not going to be wasteful. We actually carry out a recycling process here, so that as the gases leave the reaction vessel, we cool them down, the ammonia will condense to a liquid which we can run off, and any unreacted hydrogen and nitrogen are then pumped back into the reaction vessel. So in today's physics prep, we're going to have a little look at energy stores and those energy analysis diagrams. So what I've given you in the table here is a summary about the types of store, an example, and the related equations. So just because P2 is over, unfortunately, you can't forget the equations yet. So if you have, better refresh your memories. So our chemical store has no related equation. The thermal store is on the data sheet. So energy is mass times specific heat capacity times the temperature change. Kinetic energy, that one is going to be 0.5 times the mass times the speed squared. Our gravitational store is energy is the mass times gravitational field strength times height. The elastic store, 0.5 times the spring constant times the extension squared. The nuclear store is energy is mass times the velocity of light squared. And electrostatic and magnetic stores have no related equations. So you do need to go back and refresh your memory about those equations that we learned in the earlier physics topics here. So when we're talking about how this energy is transferred between stores, we've got 4 possible options: mechanically, electrically, heating by particles, or heating by radiation. So do remember, when we're asked to calculate transfers, if it's a mechanical transfer, work done is force times distance. If it's an electrical transfer, then it's power times time. One thing we should mention here is that energy cannot be created or destroyed. It's only transferred between stores, which is our law of conservation of energy. So do remember that if we're talking about a closed system, there is no net change in energy. So one of the things we could be asked to do is to carry out an energy analysis. So to do this, we need to choose 2 points in a process, which may be given to us in the question, identify which stores have more or less energy in them at those points, and then work out what type of transfer occurred between the stores. So if we look at one example here, so we've got our drag race. So at the start of the race, we have a stationary car with plenty of fuel and oxygen. The transfer is going to be an engine exerting a constant force, and then at the end of the race we have a moving car with less fuel and oxygen. So in terms of our energy analysis, at the beginning of our race we've got no energy in the kinetic store and quite a lot of energy in our chemical store. And then at the end of the race there's some energy in the kinetic store and less energy in our chemical. And the way we're transferring between them is mechanically. So when we come to draw our diagram, remember it's just little boxes with a line in there to show the amount of energy. So chemical store has a high line, nothing in our kinetic store box. We put mechanically on our arrow, and then our chemical store reduces, and however much you dropped your chemical store down by is how much you put in your kinetic store. Do remember though that not all energy is transferred to kinetic energy in this case. Some of it will be transferred to the thermal store due to friction, sound or air resistance. So we do need our third box on there to represent our thermal store, which as you can see increases slightly. But do remember that whatever your increase in the thermal and kinetic stores are, have to equate to the decrease in the chemical store. The last thing to bear in mind here is that they could very well ask you a follow-up question related to doing a calculation. So I've given you an example there of a drag racer starting from rest exerts a force of 4 kilonewtons over a distance of 300 meters. Calculate the work done. If the mass of the racer is 300 kilograms, calculate the final speed. So there's two things to do in there. Calculate the work done and calculate the final speed. So this is where we need to recall those equations that we've hopefully learned. Work done is force times distance. We can see our force is 4 kilonewtons, so 4 times 1,000 to give us newtons, and then times that by our distance of 300 gives us 1,200,000 joules. Second thing we need to do is calculate the final speed. So kinetic energy is 0.5 times mass times speed squared. If we rearrange that to make speed our subject, it's the square root of 2 times the energy divided by mass, and then substitute in your values, it gives us 90 meters per second. But you can see there, if you've not learnt your equations, you're going to have a blank space on your answer page. So do go over all of those equations. As always, if there's anything we've gone over in our daily booster today that you're not 100% certain on, head on over to the playlist for the relevant topic. So for biology it was B6, chemistry is C6, combined physics is P5, and the separate physics is P7 playlists.